In drawing 28, we're going to see three views of the embryo, and the top two are about at 21 days, but we're going to see two different views. This one called a sagittal cross-section, and this one is called a transverse cross-section. In this one down here, we're going to see it's a sagittal section with special emphasis on the vascular system. All we're going to see is the little blood vessels. Let's zoom in on the top two. This picture is a help in understanding these cross-sections. So here you have the human body sliced in various ways. The red plane going this way is called the sagittal plane, and that's what we're going to see in this drawing over here. And you'll notice that it goes right down the middle, and if it is in the middle, it really doesn't include either leg. So this plane here, the kind of yellow one, they've moved it over a little bit so you can catch a leg, but in that case you miss the head. So a sagittal view doesn't really give you the entire body. The plane that goes like this is called the transverse plane, and that's what we're going to see over here. We're going to see a transverse cross-section as if we're cutting the embryo like that. And then the blue one is called the coronal plane, and we're not going to see that at all on this page. Just thought I'd mention what that's called. So those are real good anatomy words to know: sagittal and transverse. And then I'd also like to point out this dashed line running down here, and this shows you where we've cut the embryo to get this. So if we'd sliced it over here, it would look different than here or here or here. So this dashed line shows you where we've made our cut to get this picture. So let's start with this picture here, and it's nice you don't have to draw it. All you have to do is color it. And so you color along with me, and we'll talk about each part as we go. And then here you're going to obviously label what the number is up here. You're going to write it, and you might want to use either a very sharp pencil or a pen with a fine point, because it's some of the words, we're going to write more than one word on the line. And also one more note before we start, you'll notice there's some duplicate numbers here. So for example, the number 8 here and here. So you're only going to have to write 8 once here, and then that's good for this 8 and that 8. So there's a few that are just on this drawing, there's a few that are on both, and then there's some more numbers that are on just that drawing. So we'll kind of zoom in on this first, and I want you to be able to see these blanks as I fill them in also though. So okay, let's start with number 1, and we're not even going to color anything for number 1. We're going to leave it uncolored, and number 1 is basically this space here. You can see there's the one around here, this little strip here, and all of this. Basically number 1 is the background of this whole thing, where this is occurring, and that would be in the endometrium, the lining of the uterus. So on line 1, let's write endometrium. If you can't remember the word endometrium, you might want to add in parentheses lining of uterus. But if you have that word down and you know what it means, then you don't need the extra note. So number 2 will be red. Number 2 are all these little capillaries over here. And these are the maternal capillaries. Anything related to the mother is maternal. So these are the capillaries that build up during the first part of the monthly cycle. The uterus builds up lots and lots of little capillaries just in case an embryo comes by and starts to develop, and these capillaries now have a use. And if they're not used, they're just shed at the end of the month. So I'm going to keep coloring here, not just the capillaries, but into these spaces. The blood goes like this, and it's more complicated. This picture's much simplified. These little spaces in and around, like here, these little wiggly areas, are a lot more wiggly in real life or in other drawings. Okay, now be careful not to obscure the lines and the numbers. If you're worried about it, you can even trace over the lines, but just kind of lighten up when you come to a line. Stay nice and light so that we can see them. Okay, we're going to color all the way around here. And there's names for these pockets, like everywhere it goes in there, it forms a little pool called a lacuna. And we're going to see the word lacuna. I'm not going to write it here. I'm just mentioning it so that when we get to connective tissue, you'll be like, oh yeah, I remember we heard that word before. And I like to think of it, the word lagoon. A lagoon is like a, area in an atoll, a coral reef. A lagoon is like a pool, giant pool of water. So the lacuna are these little pools that form they go in and form little kind of inlets, little lagoons sort of. They're called lacuna or lacunae, plural. Okay, so let's label number 2 here as maternal capillaries. And then 3, we're just going to write maternal blood. I'm not going to make you learn the word lacuna right now. Let's just write maternal blood. And once again, when we talk about blood, get your mind used to thinking blood, that's a good thing. Blood means oxygen, it means nutrients, food. Right? It's not yicky. Blood is the thing that brings the oxygen and food to the cells. So this is a good thing to have all this blood around here. This is where the embryo is going to get all its nutrition and oxygen. So now we have number 4. And before we color 4, what I want you to do is take your red again, and we want to draw in some capillaries coming in from the connecting into here. Now, it's a little more complicated than this, as we'll see in the bottom picture. In just a minute, we'll kind of zoom in on a little more what the veins and capillaries are doing. Right now, we're just going to use red. Usually, they use blue and red to show the blood that's going to the heart or away from the heart, or the blood that's got less oxygen or more oxygen. But right now, all we're concerned about is that there is a vessel of some kind running into here, because that is what is in these things. These are the chorionic villi, and they have little blood vessels running into them like this. And once again, it would be more complicated than this. It would have little tiny things going in there, not just one. But if we just go around and just draw one vessel going in, that's good enough. The main thing is to know it's there. Into all the little fingers. Remember, villi, or villus means finger. So these little blood vessels running in there. And then connect up right past that little thing that's labeled 7. Just take a line, a red line, and go in like this to know that these little blood vessels go into the embryo. Now we can color the chorion. Now 4 is pointing to like the whole thing basically. I'm going to take pink, and the reason is because it blends nicely with yellow, and then I can just color over like this. See, I don't have to color around all the little vessels. I don't have to worry about slopping over onto the red because you won't even see it. That makes it really easy to color this thing. So the chorion will be pink. The chorion is the outermost sac. Got all these bags and sacs. And let's label number 4 down here. Here, chorion. And then number 5 is pointing to the little vessels, chorionic villi with capillaries. So the villi are the little finger things. That's the villi. Chorionic villi, 1 villus, 2 villi, and I'm going to put in parentheses with capillaries. So then number 6 is basically going to be empty space, and let's just leave it white. Let's not color this. So remember, there's this empty space between the chorion and the amnion, and that's going to disappear as the embryo gets older, and by the time the baby is born, the amnion and the chorion have basically merged together, and you don't even notice the chorion. We just call it the amniotic sac, but the chorion was there, or still is there. If you look real carefully under a microscope, I suppose you could differentiate, but just visually, when a baby's born, you can't really differentiate between the chorion and the amnion. And let's make the, let's label 6 first. Let's label this the the chorionic cavity. Chorionic cavity. And before we do the amniotic stuff, let's just go back and pick up number 7 there. See, number 7, let's color this yellow because it was derived from that endoderm. It was going to be part of, in fact, let's keep going with the yellow. Let's just make 9 the gut tube. Let's make this whole thing yellow. This all came from that original endoderm. That's why we're keeping it yellow. And a little bulge came out and formed this little thing, number 7. This is one of the strangest names for parts of the body. Really weird. It's called the allantois. A-L-L-A-N. Allan, T-O-I-S. Allantois. And oddly enough, the word allantois is Greek for sausage. Yes, the highly technical scientific term for sausage. And it's just because it kind of looks like a little sausage sticking off. I don't know, whoever was naming it was in a punchy mood that day and decided it needed to be the little sausage. The allantois functions basically as a garbage bag. Right, so here in the embryo, we don't have kidneys and bladder functioning. We don't have any of the excretory system yet, but the cells are going to be making wastes. They're going to be having a metabolism and making wastes. And where do you put them? Well, you just put them in a garbage bag. Now, the allantois in a chicken or a bird egg is much larger and much more important. The allantois here is going to not be needed after a while. Once we get kidneys up and running, once we get everything in here, we got a nice placenta formed, we really aren't going to need this garbage bag anymore, because the baby's waste just can get sent out to the mother's bloodstream and go out as the mother's waste. But in a chicken egg, it's got a hard shell, and there's no contact between the embryo and the mother chicken anymore. So all the waste that build up over the couple weeks of development all have to be stored in this little bag so the waste don't poison the bird embryo. So in bird eggs, the allantosis is more significant. Here it's just a real temporary structure. A lot of these structures we're going to see, the yolk sac, the allantosis, and even some of the blood vessels that are forming, are all going to be temporary. We just have the problem of how to keep this embryo alive while the organs are developing. And once the organs are developed, then these are not so necessary anymore. So let's go ahead and label 8, the yolk sac. We've seen that before. You know what that is. And we'll talk more about that in the next drawing and the drawing after that also. So just know that's what that is. And number 9 is the rest of this little endoderm, or we're just going to call it the gut tube. And it is going to become a lot of things. It's going to become the intestines, the stomach, the esophagus, and also a lot of the digestive glands, the pancreas, the gallbladder, the things that are all attached to the digestive system. And also it's going to turn into the lungs as well. Alright, so 10 is going to be red. Right outside of the endoderm we have, as you know already, the mesoderm. Don't cover over the number 10, right? Make sure these numbers stay. You can see them. The mesoderm is going to develop into muscles and bones and blood and all kinds of things. Now I want you to add a red line right here, sometime here, but at some point there's some mesoderm that grows around the outside of the yolk sac. And let's just go ahead and put that in. And that's all I'm really going to say about it, because I'm afraid if I say a lot more I'll be in error, because I just haven't been able to find a lot of good information about it. But just so you know, there is a little bit of mesoderm covering this around the yolk sac. Okay, and let's label that 10 mesoderm, or mesoderm as some people say. Meso or meso, either one. So 11 would be our ectoderm, and it's always blue. Don't cover over the 11. And remember, the ectoderm is going to develop into nervous system and the epidermis of the skin. So 12 is going to be the amniotic cavity, and let's see, 13 is pointing to the actual line here, the amnion. So I'm going to use green. You could use purple if you wanted, but I thought let's be consistent in all 3 pictures. I'm going to go with green, and I'm going to make the amnion green, and then really lightly, I'm going to make light green this amniotic cavity. Of course, it's not really empty. All the cavities of the body are not filled with air, they're filled with fluid, watery fluid that has proteins and sugars and things floating around. Okay, so let's label 12 as the amniotic cavity, and then 13 is the amnion, the membrane itself. 14 is pointing to this whole thing. Our connecting stalk has gotten longer, long enough that we can actually call it an umbilical stalk. The umbilical stalk, S-T-A-L-K, stalk, like chalk or walk, stalk. Okay, so that's all our parts for that drawing. And we want to keep our colors consistent over here. So we go over here, kind of use the same color scheme. Okay, so let's color our transverse section. Gut tube yellow. I'm going to color it just a little bit inside, like that. Now, as we mentioned in this drawing, I'd like to add a little bit around the edge here, knowing that there's a little bit of mesoderm that covers, like that, around the outside. You kind of just connect it up like that. And then these little things here, we talked about this. These little dots are called blood islands. And they are forming little pieces of vessel. So in the last drawing, the previous page, we just saw these little dots. Now we're going to show some of them as actual little pieces of blood vessel. And remember, they form with blood cells already inside of them. So that little blood island, the cells in the center of the island will become the red blood cells, and the cells towards the outside will become the walls of the vessel, and on the very outside those cells will become the little supporting tissues, little muscle and connective tissues around. So we have complete sections of vessel with blood cells already inside of them, all ready to connect up and make it, we'll see in the next picture, these are going to make a whole little network of capillaries. So we don't need to label 8 because we already have it, yolk sac, and we don't need to label 9 because it's already labeled gut tube. And 10 we already have labeled. This is the mesoderm. Now, mesoderm is more complicated in this picture. And just go really lightly. Remember, don't cover over any numbers or lines. Okay, so this would all be kind of mesoderm, but we see some specialization going on here. And that would be, we see that remember in the last picture we had the somites forming, and the somites we're going to turn into dermis and muscle and bone. And so in this picture, where did our somite go? Basically our somite is here. This is the outer side of the somite, and it's going to become the dermis, which is the underneath layer of skin. Skin has two basic layers, the dermis and the epidermis. So part of the somite becomes the dermis, and we could actually go ahead and label that. It's what, number 20. We could go down here and just write dermis. Right here, dermis. So that's part of the somite. And another part of the somite is here, which would be 19. And that would be muscle. So 19 would be muscle. And then the last part of the somite, the interior part, became bone, and this is the beginnings of a vertebra. Now, at this point, it's not really bone. You really couldn't call it actual bone because it's not hard. This is very, very soft cartilage, and it's going to have to collect minerals to become hard bone. And that's going to happen very gradually as the months go by, and even into childhood, the bones are still mineralizing. When babies are born with very soft bones in a lot of areas. Okay, so that we're pointing to 16 here. So if we go back over here to number 16, we can write future vertebra. Now, it kind of is a vertebra, but the reason we're writing future is because it's really not hardened into bone yet. So remember, there's the spinal cord in the middle, and then the vertebra around, and together they're called the column. Now let's go ahead and make this blue. This is our neural tube, which is going to become the spinal cord. So there's 15. We can go back down here and label, there we go, 15. We can write neural tube, but we can also put in parentheses future spinal cord. C-O-R-D. So when you write notochord, it has a CH, but when you write spinal cord, it doesn't. You drop the H. It's just C-O-R-D. Kind of one of the tricky things about English spelling. And we still have the notochord right there. Number 17. Notochord is still hanging in there. Eventually it'll disappear, but right now you can still see it. And let's see, number 18, let's go ahead and do 18. 18 is pointing to the nerves. And remember, there was neural crest cells that came out. And formed the peripheral nerves. Peripheral means outside of the spine. So 18 would be, let's just write nerves. Came from the ectoderm. Okay, and now let's see where, let's go in order here. 21. 21, these little things right here are, just going to make little red circles around them. This is all mesoderm. Those are the future kidneys. And as long as we're here, let's write 22. The heart tubes. And I showed you a picture of how the heart starts out as two tubes that come together and then kind of morph into a heart shape. So 22 heart tubes there. So they're going to come together. And then we're just left with these lines here. Remember, we already talked about those. We saw these developing from the mesoderm. The ends of the mesoderm split. Remember, they split and started going two different ways. And one of them followed the gut tube, basically. And the other one went like this. And they are going to form the membranes that are covering the organs. Remember, we looked at this picture. We said that all your organs are in bags. Everything is like double and triple bagged inside the body, and you have a bag that goes around the organs like this, and then there's an outer bag, and the inner bag, 23 is pointing what's going to be the inner bag. And since it's going to be around the organs, we're going to use the Greek word for organ. They called any organ that was inside the chest, you know, the heart, the stomach, the liver, the kidneys, the intestines, they would call that a viscus. That was their word for organ, viscus. And from that word we get visceral. So we're going to call this number 23 the visceral, V-I-S-C-E-R-A-L, visceral. And then the fancy word for this membrane is peritoneum. Peri, P-E-R-I, and that means around, goes around, wraps around. Peritone, T-O-N-E-U-M, peritoneum. So this is a word, especially visceral here, to not be afraid of. Just remember, a viscus is the Greek word for an organ. So anything that's visceral has to do with basically your guts, anything inside your body cavity. So the visceral peritoneum is the membrane that wraps around your organs. And then this other one here is going to be the parietal, P-A-R-I-E-T-A-L. And if you studied the brain at all, you know that there's the parietal lobe in your brain. And oddly enough, the word parietal just means wall. So it makes sense here. It doesn't make quite so much sense in the brain, but here we've got the wall membrane, peritoneum, just the same, peritoneum. And the reason it's called the wall membrane is because it's attached to the wall of your body. If you think of your skin as being your outer wall, this would be what's lining the wall of your body. It's kind of like maybe the lining on a coat. If you have a fancy jacket, you know, a dress jacket, you know, for guys, you know, tuxedo, or I mean, ladies can have dress jackets too. You know, a lot of times there's like a silky lining on the inside of the jacket. That's a lot like the parietal peritoneum. Now all we need to do is finish up just a little bit of coloring here. Let's make this, or this ectoderm or epidermis. We could call it epidermis now. This is actually going to function as epidermis. See, it's already split off. The ectoderm is before it really differentiates into nervous tissue and epithelial tissues. So what's pointing to that? Number 11. So over here, ectoderm, we could add a little note, ectoderm / epidermis in that drawing. So we're actually ectodermis now. And then let's take the green, or whatever color you made your amnion. Color the membrane darkly, and then the space very light. So now we're going to take a look at just one aspect of the embryo. Now, I've said this before several times, I'll say it again, these drawings are highly simplified, and they're designed to show you particular things about the development of that stage. If we put everything in accurately, just like it looks in real life, it would be so complicated we wouldn't be able to tell what you're looking at. So the goal of an illustration is to show you a particular thing. Here we really wanted to focus on what was happening to these things, right? So in this drawing, what we're focusing on is just what's happening to the blood system, the vascular system. So here we see the embryo. You see this is the embryo, and we're not going to see the notochord and the somites and everything like that. All we need to know in this drawing is that the embryo is here, and we're just going to focus on these blood vessels. And this is the yolk sac right here, and here's the umbilical stalk. So there are other things in this picture, right? You say, well, where's the little future kidneys and where's those little nerves? We're just not showing them. So take your red and let's start to get us oriented properly. Let's start right here. See these little arch thingies? We'll color those in. We'll call it aortic arches. Follow this around. I'm going to color that one. And then it was going to lead you to this little lumpy thing here, which is actually the developing heart. And what you want to do is color the bottom part, but like only halfway, right? Just to that where that bottom line is, like that. Just leave it like that. And you'll see here that our heart tubes have already merged and gotten together to form this lumpy mass in the middle that is going to become the heart. And right now it just looks like two bumps, but it's going to fold and twist and bend in here to actually make the heart the way we see it in a newborn baby. However, it doesn't wait to start beating until it's completely formed. Even at this stage, these cells, they know that they're cardiac cells. And remember, they have those gap junctions, they're communicating, and they know that they're going to have to beat for many, many years to come. And they go ahead and start squeezing and beating, even at this stage here. So let's follow this vessel back. And this is the vessel that goes to nowhere, right? It doesn't really have anywhere to go yet. It will, but right now it's just kind of waiting. Let's follow it to where it goes behind here. And you'll see this is labeled number 5. And then down here like this. This is where it's going to get interesting. Follow it like under and over. It's going to squiggle around a little bit as it goes out into the chorionic villi. So just kind of keep following over and under like that. And then it's going to go like this. Okay, now just leave it like that for just a minute. Let's go back in here and finish. This also goes down this way. Color that little thing. And that. Down to here, and then just pause. We'll come back and we'll catch that in a second. Switch to your blue. And then all the other little vessels are going to be your blue. There's another vessel going to nowhere. You know, sometimes this reminds me of roads that I've seen in places where I've lived. I've noticed, I've lived there long enough that I've been able to observe the roads over the years, and sometimes they would put a road out that they would think would be going to a new housing development or another highway, and then they get the road built because they knew where it was going, but it would sit for a couple of years before they finished whatever else they were doing here. So it would kind of be like the road to nowhere, and eventually then they'd come back and they'd connect up, but the end of the road just kind of went into a field or something. That's what it reminds me of. Go around this way. And now we're going to do the top of that heart area. Remember, don't cover over those little lines. and then down into the yolk sac. And then pick it up over here where we kind of went through the umbilical stalk and out. We'll just finish all the coloring and then we'll label on this one. Now we're going to finish this area and then these. So you want to take either red or blue. I guess as long as I'm holding blue, I'll start with blue. And make a line that comes down here like this. And then you're going to make little lines only to the halfway point. I've got a few started for you, just a little like network here, like a little intertangled little bunch of capillaries. You've probably seen pictures of capillary networks, how complicated they are. Right? And it doesn't matter exactly how yours look. They don't have to look exactly like mine, something like that. And then of course on the other side with the red, you're going to do the same thing so that they match up and eventually a red line will meet a blue line. And usually in anatomy pictures, when you see this, there's like these little capillary beds and all over your body where the teeny tiniest capillaries come through, and this is where they're the smallest individual blood cells go through them. They're very, very tiny. They're microscopic. And usually they use red to represent oxygen-rich blood, and this would be the point at which the oxygen gets delivered to the cells, and then there isn't so much oxygen, so then they use the color blue to represent less oxygen, so these would be the veins starting back, going back to the heart. And you can see here, like if we travel going this way, the blue, we could make it go back to the heart, and then let's say the heart pumps out red again, and the red, but first it's going to have to go like this, all the way through here. Somewhere out in here, it'll turn blue, and then come back through the blue network again. So let's make the little network out here, red and blue. So you've got one of each coming to each of the villi here. So the easiest way to do this, if I can zoom in, I'm going to start with my red. Go like this, make a loop up like this, and then stop. And then pick up your line down here again, the red line, and go into the next loop and stop. And then connect up again like this. So you've got kind of these three red hooks going up. And then down here the same thing, up into this one, like that. Then with your blue, pick up where you left off on the red line. Now you may have to kind of go underneath that red line to come back to the blue line like that. And you want to do that with all of them, making a blue line, connecting up to blue. There. Now if we try to follow this all around as if it were an adult body, it really kind of wouldn't make exact sense because we don't have any lungs yet. The job of our heart is to pump the blood into the lungs, and then the blood comes back to the heart, and then the next round it goes out to the body, but we don't have any lungs here. What we have is a yolk sac and some chorionic villi. So it's kind of a strange loop, you know. So you'll start with the heart. the blood going away from the heart is going to be red, so it goes away, away, goes to nowhere, and then around here. It could come down here, or see it splits. It could come here, and it could go out, and remember the mother's blood would be on the outside here, so we could get some gas exchange. We could get a little oxygen coming in through here, and then it would come back in through the blue. It would switch right here, red to blue. Whoops, now we're on the blue track. And then it goes blue, 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 coming into here. And it could go back out into here right now, or it could go down in here and then go back out, or it could go through here. So it's got some funny options here that really aren't going to be available to the blood in a few months. Once this heart and lungs are in place, this type of circulation scenario here, it isn't going to exist anymore. And some of these vessels, we can't say, oh, exactly, here's an aorta. I mean, well, it sort of is, but some of these are temporary, and they're going to be shut down. And it's like the detour. You know, you build an extra road on the side while something's in construction, and then you don't need that extra road anymore, and so you'll take it off. So a few of these are detours that will be dissolved and gotten rid of. So the main thing to notice, though, is that we now have a little heart. This is by 26 to 28 days. The heart tubes have joined. We actually have a heart starting to grow. It doesn't have 4 chambers yet, but it will in a couple weeks. And what's happening here, most likely, is that there's some nutrients inside the yolk sac. Not a lot, but just enough. This is a critical stage of development where we're not getting a lot of nutrients here yet. And that original stash of nutrients that the zygote was running on are running low. So somehow or other, and I couldn't find a good explanation for it, they just say, oh, it derives some kind of nutrition. It never tells you where the nutrition came from in here. Is this all still from the original zygote, or I'm not sure how the yolk sac is providing nutrition, but there's some kind of nutrient value in the yolk sac, and this is where it's being picked up. The blood's running through these and absorbing the nutrition and then taking it back into the embryo. And here we hopefully have a little bit of oxygen, carbon dioxide exchange going on. And in this picture, what don't we see? We don't see the allantois. We don't see the somites. We don't see a lot. Well, let's go ahead and make the embryo color. I'm going to use like pinkish or kind of like a peach color or pink or something light that will just sort of distinguish this from this, so that we can see what's embryo and what's not embryo. And down here it's kind of ambiguous of where actually the embryo stops and the umbilical stalk starts. So around here, this would be embryo. And then I'm going to make the yolk sac yellow, just because we have been. And remember, there'd be a little bit mesoderm, but we don't really need to show that right now. That's good enough. Just yellow for that. Somewhere the allantois would be down there somewhere. And then this is the amnion. I'm going to use green because I have been, to make the amnion green. And then I'm going to do the same trick where I'm going to shade real lightly the inside here, so it's consistent with our top drawings. That's the amniotic cavity. So now let's label. Number one is this band going all the way around. So this will be the chorion. Number 2 are these things, which are the chorionic villi. Number 3 is pointing to the yolk sac. Number 4 is pointing to this blue vein, and this is called the cardinal vein. Cardinal, just like the bird. And the word cardinal, the meaning of the word means important, foremost. And the bird cardinal, the cardinal bird, is red, and it's called a cardinal because it reminded people of the cardinals in the Catholic Church who wore red, and they were called cardinals because they were the most important, like right under the Pope. They were right up there. The cardinals were at the top, so they were very important. So cardinal means important. So we've got the super important vein right there. And number 5, pointing to the red one running behind, and that is called the dorsal aorta. Dorsal, you should know by now, means back, and aorta is what you call really large veins coming out of the heart. And we'll see that word again when we study the heart. So that is the dorsal aorta. Number 6 is just pointing to this whole thing. It's the embryo. And 7 is the amnion. 8 is the amniotic cavity. 9 is the future head/brain. That's the way we're looking at it. So the embryo is like this. Okay. And then 10 is the future heart, or the growing heart. It's actually a little heart now. And you could put a note, has begun beating. All the cells are contracting in rhythm together already. So it's functioning as it's developing. And the very last thing I want to draw your attention to is a note right here, and we have one right up here also, where it says actual size. So how big is this thing that we're looking at? Obviously we've drawn it a lot larger than real life. It says actual size is 2 millimeters. The embryo, this part here, would fit into this letter O. See that tiny letter? It would easily fit into there. And up here, just half a week earlier, the size is 1 millimeter, and the embryo would fit into this letter O. That's even smaller.